Hello everyone. The topic that we are going to study today is biomolecules, which is the last chapter of organic chemistry in class 12. Again, very interesting chapter. So let us understand here. So first, what we are going to study about is carbohydrates. What are carbohydrates, right? So you must have studied about carbohydrates in your younger classes, like glucose is a carbohydrate, right? But what are these? These are hydrates of carbon, right? Okay, so let us see. What is the general formula? The general formula is CXH2OY, right? So, for example, X and Y can be like any number. For example, here the X is 6 and again the Y is 6. So, we are getting C6H12O6, which is a glucose. And we know that glucose is a carbohydrate, right? But then there are certain exceptions as well. So, we cannot totally rely on this formula because if you see acetic acid, it is not a carbohydrate and it fits into the formula very well. C2 H4 and O, like O2. So basically CH3COH, but it is not a carboxylic acid. So we cannot totally rely on this formula for, identifying if something is a carbohydrate. So we cannot use this formula, right? Like rhamnose is there. Rhamnose is a carbohydrate, but then rhamnose does not follow this formula. So this formula, we do not follow it, right? So reaction have shown that carbohydrate, they contain specific functional groups, right? So let us see. Carbohydrates are also known as saccharides, right? Okay. So let us see again another type of division of carbohydrates. So carbohydrates, what do we call them? Now we cannot relate to carbohydrates with that formula that we saw in the last slide. We call them optically active polyhydroxyaldehydes or ketones because there are certain functional groups like there is aldehyde functional group, there is a ketone functional group in the uh, carbohydrates, right? So if you see here, yes, like sucrose, right? So the sugarcane juice, sucrose is present, right? So sweet carbohydrates, we call them sugar, right? Sucrose in sugarcane, right? Okay. So milk, right? Milk. The milk, there is lactose sugar present in milk, right? So again, we call it a sugar because there is a sweet carbohydrate. Okay. So next, we are classifying them on the basis of their hydrolysis, right? So basically, hydrolysis means breaking it. So if I break a large carbohydrate and if it is made up of only one unit, I call it monosaccharide. If it is made up of like 2 to 3 units, I call it oligosaccharide. But if it is made up of many units and it gives many saccharide units on hydrolysis, that means we call it a polysaccharide. Next is reducing or non-reducing sugars, classification on the basis of the reducing nature, right? Okay. So next is like, as I told you, on hydrolysis, it cannot be further hydrolyzed. It gives only, 1 unit and it is already a 1 unit compound. So obviously it will not further hydrolyze or break to give polyhydroxyaldehydes or ketones, right? Like glucose is there, fructose is there. They are 1 unit. So they cannot break down to give further units. Now, what are oligosaccharides, which on hydrolysis yield 2 to 10 units of monosaccharides, like sucrose is there, maltose is there, right? So when we do hydrolysis of sucrose, we get, glucose and fructose, basically 2 units. So we will say that sucrose is a oligosaccharide. What are polysaccharides, which yield a large number of monosaccharides on hydrolysis? That means hydrolysis means breaking with the help of water, like starch. If I break starch, I will get n number of, monosaccharide units. So this is the type of classification of carbohydrates: monosaccharides, oligosaccharides, and polysaccharides, right? Like cellulose, glycogen, these are all examples of polysaccharides, right? Okay. Now, the next topic that we're going to study is proteins, right? So we are going to cover carbohydrates, proteins, and nucleic acids in very brief today. And again, if you want to study in detail, we can connect to Urban Pro. We can have one-on-one classes. Let us see. So protein, basically, it is derived from the Greek word proteus, which means of prime importance. So as you all know that for growth and repair of body, proteins are needed, right? So let us understand what are the normal, sources of protein. Like we get it from cheese, pulses, peanut, right? Milk, again, fish, chicken. So these are all sources of proteins where we get proteins for our body. Okay. What are proteins? How are they made up of? Basically, they are made up of alpha amino acids. This chapter is basically also linked to amines, right? They're made up of amino acids. What are amino acids? Where you have amine and you have carboxylic acid group as well. Basically, if we link many amino acids together, we are going to get protein, right? So there are acidic group, there's a basic group, right? This is alpha amino acid. Now, what is a beta and gamma amino acid? Let us see. So this is alpha amino acid because, as I told you before also, what is the alpha carbon? Alpha carbon is a carbon which is next to the functional group, right? So this is the alpha carbon, also it is next to the functional group, right? And here the amino group is attached to the alpha carbon. Here it is attached to the beta carbon because it is second in position with the COH functional group. So beta carbon, right? So amine NH2 group is attached at the beta position, right? So this is beta amino acid. Now if it is attached at the third position, this is alpha, this is beta, this is gamma. So we are going to call it gamma amino acid, right? Okay. So when these amino acids join together, they form a long chain of compound that is known as protein, you know. So let us see the representation of amino acids, like how amino acids are represented. So we have this, and if I replace this with another amino acid group, what will happen? New amino acid will form, right? It can be any group, right? So for example, can you see here? I replace, this is the basic amino acid, which is known as glycine. The symbol is GLY, right? And the code is G. Here I replace it with this, right? So this is known as triosine. Basically, amino acid is nothing but which has NH2 and COH. So different type of amino acids represent different proteins, right? Okay. Now, how do we classify the protein based on their shape and structure? So there are primary structure of protein, secondary structure, tertiary, quaternary. Secondary is again divided into alpha helix and beta pleated. Let us see. So amino acids are linked together in primary structure. They are linked together in a specific sequence, right? Can you see how they are linked together in a specific sequence? Okay, these are primary amino acids. Okay, so now this is secondary amino acid. So basically there is this chain of amino acids and there is a regular folding in the chain of amino acids, right? So it twists, right? So it twists like a right-handed helix. Why does it happen? This is because of in the amino group, like if I have the okay, NH2C, C double bond OOH. Basically, there is a hydrogen bonding between this NH group and the, this oxygen atom, right? Which makes it twirl, right? Which makes it twirl. So that is the reason these are secondary amino acids, right? So as I told you, alpha helix, this is alpha helix, which is a secondary amino acid. Okay, that is secondary protein, sorry, beta pleated. So it forms a pleated sheet like a drapery. So you can see here, so we have this NHC double bond O, right? Okay, which is again, so NHC double bond O. Can you see NHC double bond O is there, right? Which is again linked together. Many amino acids units are linked together to form the secondary structure of protein, right? So it forms like a sheet, like a drapery sheet, a pleated sheet. So we call it a beta pleated sheet, which is again a secondary structure, which is held together by intramolecular hydrogen bonding. So can you see there are two sheets which are held together by intramolecular hydrogen bonding? We have hydrogen, we have oxygen. Again, oxygen, hydrogen, they are joined together by intramolecular hydrogen bonding, right? Okay. Okay. So basically how these are bonded, let me show you. So I have NH and C double bond OOH, and there is okay NH2 C double bond OH, and suppose done it. Yes. Okay, suppose if I have NH2 C double bond OH plus again I have here NH and C double bond OH. So basically here these H2O molecules they get removed to form water molecules. So you get NH, C double bond O, NH. So you can see NH, C double bond O, NH, right? And there can be any also number of carbon atoms in between, right? So this is the type of bonding basically in case of the secondary structure of proteins. So let us move forward. Now tertiary structure. Now basically primary if it is folded further secondary. If I fold the secondary structure again, so I get a tertiary structure, right? So now this is giving two major shapes like fibrous and globular, right? So sometimes in exam they ask us what is the difference between fibrous and globular. Let us see. Now how does this structure become stable? Because of the hydrogen bonds, disulfide linkages, there are van der Waals forces, there are electrostatic force of attraction. Now Fibrous and globular means globular means it is like a circular in structure. Fibrous this means like it is just like fibers, like strands of hair, how the hair are fibrous, right? Okay. Next is quaternary structure. Basically, if I twirl it more, if we, if we if we make it much more complex, we get quaternary structure of protein. It is composed of, more polypeptide chains together. It is a very complex structure. Let us represent all four types of protein. So can you see, this is primary, which is very simple. So we twirl it again. Again, if you make it much more complex, it becomes tertiary. You make it much more complex, it becomes quaternary structure of protein. Now what is denaturation of protein? Basically proteins, if we change temperature, pH, so protein, they lose their biological activity, right? So this is a functional protein. So if you change the temperature, if you change the pH, the protein loses its function and it does not work now, right? So can you see here, the globules have unfolded. So now the protein is ineffective if you change the temperature, right? So let us understand the, some of the daily life application of this. So you must have seen about the egg yolk, right? So if you boil the egg, what happens? The proteins actually they become denatured. That is the reason. That is the reason that the egg, the proteins, that they are not no longer functional now, right? Okay. Next is nucleic acids. What are nucleic acids? Let us understand. So basically here we're going to talk about genes. So a father and a child, they look very similar. Again, a dog child and the dog's mother, they both look similar. Why? Because of the genes, right? Because of the genes that are being transferred. Okay. So these genes which are present in chromosomes, they are responsible for heredity, and again they are made up of proteins and nucleic acids. What are nucleic acids? There are two types, that is DNA and RNA. DNA is deoxyribonucleic acid, RNA is ribonucleic acid, right? And what are nucleic acids? They are long chain polymer of nucleotides. So we need to understand what are these nucleotides. Okay. So nucleic acids are also known as polynucleotides. Okay. So let us consider what is the chemical composition of nucleic acid. One nucleic acid, it is made up of, pentose sugar, phosphoric acid, and nitrogen containing bases. Let us see now. Okay. So sugar like this, there is a pentose sugar like ribose. So we have D-ribose here. And there is, again, this is another sugar, that is deoxyribose. So you must have heard about RNA and DNA. They are all nucleic acids. You know what is the full form of DNA? It is deoxyribonucleic acid. So this, it is a nucleic acid. It is RNA, ribose nucleic acid. RNA is made up of beta D ribose. DNA is made up of beta D2 deoxyribose. Now, if you would see, as I told you, nucleic acids, the composition is sugar, phosphoric acid, and a base. So this is the sugar, this is the phosphoric acid, and we have the bases, nitrogen-containing bases, again, which are divided into two types, that is purines and pyrimidines. Purines are adenine and guanine. Pyrimidines are cytosine, thymine, and uracil. They are combined together to make the nucleic acids, right? So basically, sugar used is beta-2 deoxyribose or beta-D ribose. We have phosphoric acid and bases. These are the bases, right? Okay, so suppose we have beta-2 deoxyribose and I mix phosphoric acid and I have adenine. So adenine and guanine pair together, cytosine and thymine pair together. Now, what are they going to make? They are going to make together a nucleic acid, which is DNA. I hope you understood this. This is very important. It is asked in the exam, adenine is paired to which base, cytosine is paired to which base, right? And what is nucleic acid? Basically, DNA made up of. DNA is made up of three things. Nucleic acid, that is a sugar, phosphoric acid, and a base. And these are the four bases that are involved in the formation of DNA. So you can see here these bonds, these are hydrogen bonds, which are linking this adenine and guanine, cytosine, and thymine, right? Okay, let us see RNA, how RNA is composed. So these are the sugars, phosphoric acid, and bases we have studied about. If we have beta-D ribose, if we have phosphoric acid, and if I have adenine, adenine always bonds to guanine. Now, there is one difference between DNA and RNA. Again, the first difference is between the sugars, and the second difference is here. Cytosine is there. Now, it does not bond to thymine, but instead it will bond to uracil. So this together makes the RNA, right? That is the nucleic acid. So I hope you understood what all we did today. So we started biomolecules with understanding about carbohydrates. Then we moved on to proteins. We saw about primary, secondary, and tertiary. Although we covered it in brief in a very small amount of time, And then we moved on to nucleic acids, right? We saw how are nucleic acids made, nucleic acids like DNA, right? DNA is deoxyribonucleic acid. What is the sugar? What are the bases involved in DNA? Similarly to DNA, RNA, what is the sugar? What are the bases involved, right? So here we have covered everything in brief in a very short amount of time. If you want to learn about biomolecules in detail, you need to practice board questions and understand this chapter. So we can do one-on-one sessions on Urban Pro. We can connect through Urban Pro, right? So thank you so much. Hope you liked this session. Thank you for your time and attention.